In this video, we are going to continue talking about catabolism, specifically looking at stage 4, which involves the electron transport chain. Before we look into that more deeply, let's recap what we have talked about so far. We looked at the molecule ATP, which is going to be our energy-carrying molecule, and we looked at how its hydrolysis can be coupled to reactions that need energy, because the hydrolysis of ATP releases a lot of energy. And again, making ATP is the whole point of catabolism. We want to have this molecule around in order to fuel all the reactions that need that energy. We also looked at reduced coenzymes. The reduced form of the coenzymes of interest are NADH and FADH2. These are the molecules that are going to feed into the electron transport chain. And those are the molecules that are produced in the citric acid cycle, which we also talked about. We talked about the specific reactions that produce these reduced coenzymes along with this carbon dioxide. So now we are ready to look at the electron transport chain and how it is that NADH and FADH2 feed into this chain in order to ultimately produce our ATP molecules. So before we look at the details of the electron transport chain itself, let's orient ourselves again in the anatomy of the mitochondria. So first, let's recall that the matrix is where the citric acid cycle happens, which I can also refer to as the TCA cycle. Okay. So this is where all of those reactions are happening. This is where the NADH and FADH2 are being produced. So again, recall that your mitochondria has two membranes, right? It has this inner membrane shown in yellow, and then it has this outer membrane shown in brown. The matrix is what is housed inside that inner membrane, and again, that's where the TCA cycle reactions are happening. Now, the electron transport chain is going to be embedded in this inner membrane here. So this is where the electron transport chain is located. What I mean by embedded, it means that the enzymes that compose the electron transport chain, which we're going to look more closely in the next slide, those enzymes are going to be embedded in this membrane so that some portion of the enzyme accesses the matrix and then the other portion accesses the intermembrane space. So the intermembrane space is going to be an important part of the electron transport chain reactions, because some of the products that are going to be produced by the enzymes in the electron transport chain, some of those products are going to accumulate in the intermembrane space. Those products are going to be protons. And this accumulation of protons within this intermembrane space, that accumulation here is what's going to be the important part of driving the phosphorylation of ADP into ATP. So now with this in mind, let's go ahead and look at the details of our electron transport chain. So here I'm showing you a schematic of what the electron transport chain and ATP synthase look like. Okay, so first you're going to see that there is this first complex here titled with Roman numeral 1, Roman numeral 2, 3, and 4. So this would be complex 1, complex 2, complex 3, and complex 4. These are all enzymes, and they are going to carry out certain reactions. So before we look at the specifics of these reactions, let's orient ourselves in what is being represented here. First, let's look at the matrix. So all of the reactions down here, which include NADH and FADH2, these are happening in the matrix of the mitochondria, which again is on the very inside of that double membrane. Then we can see that this membrane here is going to be the inner membrane. So it's the one shown in yellow in the schematic here. And then on the other side, we have our intermembrane space. And you can see that our protons are accumulating in this intermembrane space. So what this means is that keep in mind that there is another membrane here that's not being shown. This lipid bilayer, this other membrane, is going to be what we refer to as the outer membrane of the mitochondria. So this means that on the other side, over here, we have the cytoplasm of the cell. So just keep in mind that these protons here are being enclosed in the space in between the outer membrane and the inner membrane of the mitochondria. All right, so they will accumulate in this space. So first, as already mentioned, there are going to be 4 enzyme complexes that are part of your electron transport chain, along with one more enzyme, which is this one here, ATP synthase. This ATP synthase is going to be what converts ADP into ATP. And this here, again, is our goal. Now, there's going to be some terminology that is used interchangeably. Sometimes electron transport chain and oxidative phosphorylation is used interchangeably to mean the same thing. basically to mean all 4 complexes shown here with Roman numerals, plus your ATP synthase. And so sometimes those two will be used interchangeably. However, sometimes oxidative phosphorylation is used to refer to just the ATP synthase, and the electron transport chain term is sometimes used to refer to just the first 4 complexes. But in this video, we are going to use electron transport chain and oxidative phosphorylation interchangeably, and it's going to mean all of the 4 complexes and the ATP synthase. All right, so with that out of the way, let's go ahead and look at the reactions that are happening here. So what is feeding into your electron transport chain is, again, your reduced coenzymes. So NADH is going to feed into complex 1, and FADH2 is going to feed into complex 2. So since all these complexes are enzymes, this means they are going to carry out reactions. The reactions they're carrying out is the oxidation of NADH to NAD+, and the oxidation of FADH2 to FAD. So when this oxidation reaction happens, there are 2 important products here. One is that this proton is going to be pumped across this membrane into the intermembrane space. The other thing is that there are going to be 2 electrons that are generated that are going to be shuttled off to complex number 3. If we look at complex number 2, the same thing is going to happen with FADH2, where these 2 electrons are produced and they are shuttled over to complex number 3. However, you will notice that for FADH2, there is no proton that is being pumped across the membrane from the matrix into the intermembrane space. All right, so at this point, we have oxidized NAD+ and pumped a proton across. We've also oxidized FADH2, and both reactions have produced these electrons that are being shuttled over to complex number 3. Now, this transfer of electrons here is going to help pump another proton into the intermembrane space. Now, these electrons are going to continue to be shuttled over into complex 4. And at this point, this is where the shuttling of electrons stops, and that those electrons then help reduce oxygen to water, along with pumping another proton into the intermembrane space. So this reduction of oxygen to water is what makes us aerobic. Any animal that does this kind of ATP synthesis is going to be an aerobic organism. All right, so from here, the thing to note is that we are accumulating protons in the intermembrane space. The proton concentration in the intermembrane space is going to be higher than the proton concentration in the matrix. It is going to be much lower in the matrix. And it is this accumulation of protons that is going to drive the synthesis of ADP. So these protons here in the intermembrane space are going to want to flow back into the matrix to equalize this concentration difference. And it is this flow of protons through this ion channel. So you can think of there being as a little tunnel through this enzyme, this ATP synthase enzyme, where these protons are going to flow through there. And that flow is going to provide the energy to phosphorylate ADP and convert it to ATP. So this is the final step of our electron transport chain and the final step of catabolism as a whole. And this is where most of the ATP is generated. All right, so now let's apply all of this information to filling out a diagram like this. I want to note here, we are going to be using this information. I will be reiterating it again in a separate problem where we can calculate how much ATP or how many ATP molecules we can make from NADH and FADH2. So NADH, as we already mentioned, feeds into complex 1. And when it does this entire shuttling of electrons and pumping of protons, at the end of the day, will get you 2.5 ATP molecules per NADH molecule. For FADH2, that one feeds into complex 2, so it will produce slightly fewer ATP molecules per molecule, but it's still 1.5 molecules per FADH2 molecule. So we will use this information to solve some problems later in this video. All right, so let's apply all of the information we just discussed. We're going to apply it to this empty schematic in order to just try to recall everything we just talked about. All right, so first let's orient ourselves. We see that we have the matrix down here. The outer membrane is this one here. So that means this is going to be my intermembrane space. and my inner membrane is where all of my complexes are embedded. So this is going to be my electron transport chain, complex 1 through 4. And then this last complex here, therefore, must be my ATP synthase enzyme. So I will write it here. So this is going to be my ATP synthase enzyme. And again, ATP synthase takes care of synthesizing ATP, which means that this compound G must be ADP because ATP synthase converts ADP into ATP. So let's look at the other missing information and start filling it in. Okay, in complex 1, we talked about how in complex 1, NADH is the one that feeds in at this point. So NADH comes into complex 1, and complex 1 will oxidize it into NAD+. We also talked about in the previous slide that in complex 2, this is where FADH2 comes in. So FADH2 is denoted here by the letter C. And then complex 2 will go ahead and oxidize FAD. Okay. So now recall that for complex 1, when NADH is being oxidized, there are protons that are being pumped across. So I will write it in like this to remind ourselves that that complex will pump protons across. Now recall that complex 2 does not pump any protons across, but it does produce those electrons that are important for the reactions in the electron transport chain. So there's going to be electrons from complex 1 that shuttle over to complex 3, and also electrons from complex 2 that will shuttle over to complex 3. Now those electrons that arrive at complex 3 will also shuttle over to complex 4. And that shuttling is going to, again, allow protons to be pumped across this inner membrane into the intermembrane state. All right, so then lastly, my complex 4, this is where the electrons arrive, and it's what helps reduce oxygen to water, along with pumping more protons across that inner membrane. All right, so this is basically everything we talked about in the previous slide. So now let's answer some questions about the electron transport chain or oxidative phosphorylation. All right, which protein complexes are transporting the substance represented by black dots into the intermembrane space from the matrix? So these little black dots here, these are representing basically H pluses or protons. Okay. So which protein complexes are the ones responsible for pumping these protons? Well, that's going to be complex 1, complex 3, and complex 4. So this is to note that complex 2 does not pump any protons across the inner membrane. Right, now we're going to write the symbols of the protein complexes from which electrons, so now we're talking about electrons, not protons, are being transported away. So what this means is if we look at our complexes, we see that complex 1 has electrons that are being transported away into complex 3. So complex 1 is one of those. Complex 2 also has electrons that are being transported away into complex 3. And then there are also electrons here that are being transported into complex 4. But that transport of electrons stops at complex 4, so this would be the answer to that question. Number 3, what is the enzyme labeled S? Well, that's going to be our ATP synthase. And lastly, is substrate C, so substrate C is going to be this FADH2. Is it being oxidized, reduced, or neither? Well, this is going to be oxidized because FADH2 is being converted to FAD. So the answer to this is oxidized. Great, so these are all of the details of the electron transport chain. But now, remember that our goal was to produce ATP. So let's put some numbers to this. Okay, so now we can talk about the ATP yield from oxidative phosphorylation, which again means just all of the reactions in complexes 1 through 4 and that ATP synthase. All right, so we can put numbers to this. In one turn of the citric acid cycle, okay, we can produce a total of 10 ATP. So let's see why that is. All right, so recall that in one turn of the citric acid cycle, we produced 3 NADHs, and that is being shown here. And then recall that we mentioned that for every NADH molecule, since it feeds into complex 1, we can produce 2.5 ATP. So this means that in one turn of my citric acid cycle, I have 3 NADHs times 2.5 ATP per NADH, that gives me a total of 7.5 ATP molecules that I can produce from just my NADH reduced coenzyme. Now for FADH2, we only produce one of these per turn. And then each one of these molecules can produce 1.5 ATP molecules in the electron transport chain all the way up to ATP synthase. So that means that my FADH2 is going to contribute 1.5 ATP molecules once it is oxidized by the electron transport chain. Then lastly, we talked about how there is one GTP molecule that is produced in the citric acid cycle. GTP is readily converted to ATP in the cell, and so that will contribute 1 ATP for a grand total of 10 ATP. And again, this is 10 ATP per one turn of the TCA cycle. Okay. Now let's answer some questions. A certain nutrient molecule X is digested, and the products of digestion enter the citric acid cycle, which ultimately produces 4 carbon dioxide molecules. So immediately we know that in one turn of the citric acid cycle, one turn will produce 2 carbon dioxides. So if we're producing a total of 4, well, 4 divided by 2, that's going to give us 2 turns. So here we are having 2 turns in the citric acid cycle, each turn producing 2 carbon dioxides. All right, so now the question is, how many ATP molecules are ultimately produced? Ultimately means once it goes through the electron transport chain and oxidative phosphorylation. So ultimately produced by the catabolism of X. Okay, so here, let's go ahead and start by writing down our molecules of interest. Since we are interested in calculating the amount of ATP molecules, that means we need to consider NADH, FADH2, and GTP, which is made in the citric acid cycle. Carbon dioxide is also made in the citric acid cycle, but the carbon dioxide will not produce any ATP, so we're not going to consider that here. Okay, so let's just recall from the previous slide what happens in one turn of the citric acid cycle, and then we will convert it into 2 turns, since that is what's happening here. Okay, so in one turn, let's talk about the number of molecules of each NADH, FADH2, and GTP that are produced in one turn, the number of ATP molecules associated with each one, and then we'll calculate the total ATP, again in just one turn. So we're just reiterating what we discussed in the previous slide. Okay, so we can make 3 NADH in one turn of the citric acid cycle, 1 FADH2, and 1 GTP. We mentioned that for every NADH, you make 2.5 for a total of 7.5 ATP. For FADH2, that generates 1.5 ATP molecules for a total of 1.5 in one turn. And then this GTP will just be converted straight to ATP. So that will produce 1, and this gives you a total of 10 ATP. All right, so we discussed that here we don't have 1 turn, we have 2 turns. So to calculate the total of ATP, we would just multiply this by 2, because we've done 2 turns, and then that will give me 15 ATPs for the total NADH molecules. I will have 1.5 times 2, that gives me 3, and then 1 times 2, that gives me 2. And this is a total of 20 ATP molecules. So this is how you would calculate the total amount of ATP molecules that you can produce, depending on how many turns you generate or you go through in the citric acid cycle. Okay, lastly, let's go ahead and answer these questions in order to summarize all of the catabolism information that we have covered so far. Okay. So in what stage of catabolism do each of the following processes occur? Right, so here, conversion of a monosaccharide to acetyl-CoA. So acetyl-CoA, again, is that molecule that is feeding into the citric acid cycle. So that acetyl-CoA is the product of stage 2. So if we look at our roadmap here, we see we have monosaccharides here. And this is being converted to acetyl-CoA, which is right here. So this means that this is happening in stage 2. And now the question isn't asking this, but I'm just going to write it in. Here, that process is happening through the metabolic pathway called glycolysis. So that we become a little bit familiar with these other metabolic pathways in this roadmap. Okay, so glycolysis is going to be the process or the metabolic pathway by which monosaccharides are converted to acetyl-CoA. It could be hydrolysis of a triacylglycerol with lipase. So a triacylglycerol we know is a lipid, and it's being hydrolyzed into with lipase, and that hydrolysis is going to produce these fatty acids and the glycerol. So this hydrolysis reaction is going to happen in stage 1. See, reaction of oxygen with protons and electrons to form water. Well, this is exactly the reaction that we talked about in stage 4 in the electron transport chain, specifically looking at complex 4. So this is the aerobic part of respiration here, right? This is what it means to be aerobic, is that this oxygen is necessary in order for that reaction of complex 4 to happen. All right, conversion of succinate to fumarate with FAD. So we know FAD is a reactant in the citric acid cycle. Okay, FAD gets reduced when it participates in the reactions of the citric acid cycle. Okay. So because FAD is an oxidizing agent, it works to oxidize succinate into fumarate. And recall that fumarate is going to be that alkene. FAD is only reacts once in the citric acid cycle, and it reacts to produce an alkene. So this means that this happens in stage 3. All right, lastly, degradation of a fatty acid to acetyl-CoA. Again, acetyl-CoA, this is the product of stage 2. So we know this is happening in stage 2. And looking at our roadmap, we can see that the specific metabolic pathway in which this occurs is called fatty acid oxidation. So this metabolic pathway, which is a series of reactions, this is how a fatty acid is converted to that acetyl-CoA molecule that then feeds into the citric acid cycle. So with that, that will conclude this video on what the electron transport chain is, how it is that we are able to produce these ATP molecules, again, through that ATP synthase enzyme, right? And that is how we can generate from consuming lipids, carbohydrates, and proteins. That is how we can end up generating ATP molecules, which will then go on to serve as energy carrying molecules in other reactions.